Hello everyone. Hey, so this is my first official video now for my channel, and I figured today we will start with something quite simple. So today what I'm going to talk about is how to design PCR primers basically. Now these primers are only for example, if you have a sequence that you're interested in a vector and you want to make sure to confirm that's actually there. These are primers that you will use. These primers are not for cloning or anything because actually I'm going to do another tutorial, a step-by-step tutorial of how to actually use these programs to make PCR primers for appropriate cloning. So today it's just very simple, very short. So let's get started then, shall we? Okay. So for this tutorial, I'm gonna have this piece of DNA that I'm interested in. So pretend that this sequence of DNA is actually in a vector, and that we want to kind of amplify this highlighted orange area sequence, right? So this whole area, which is 1,300 base pairs, this is the piece we want to amplify. Now, how do we do that? So of course, we need to do a PCR. Now, I'm not going to talk about what PCR is and the background information because I'm going to do another video kind of on like different molecular biology concepts, and PCR is going to be one of those topics. So this is just going to be completely straight and to the point of how to design primers for PCR. So of course you know that we need two primers, right, for a PCR reaction to work. A forward and reverse. So how do we actually do that? Well, it's actually quite simple. First of all, we want to design the forward primer first. I mean, it doesn't really matter which one you do first, but mostly people start with the forward ones. Very simple. So what I like to do is using the sequence builder program, which most universities have these programs that you can use for free. That I like to construct a primer around 24 base pairs. Minimum of 20 is what I've heard and read about. That should be 20 exact base matches. But I like to go a little bit more than that, so I usually like to do 24 for standard primer design. So I have this 24 bases highlighted at the beginning, and I'm going to indicate that by using a new feature function, and I'll just type in primer. Well, I'll type in forward primer. Okay, and I can change the color, so easier to see, I guess. Okay, like that. So here we have the forward primer design. And again, it's the first 24 base pairs at the beginning of the sequence that you're interested in. Okay, so next we want to design the reverse primer. Now, the reverse primer, instead of let me back up a little bit actually. So the forward primer is based off the 5' to 3' sequence, right? Just the first top strand of this DNA sequence, the 5' to 3' sequence, which is a complement to the 3' to 5' bottom sequence. Reverse primer, as it sounds, exactly the opposite, right? So it's the So it's the bottom strand which will be complement to the top strand. In that case is where we can create, amplify this piece of orange sequence that we're interested in. So I'm just going to start highlighting from the bottom because that's what we're interested in. So again, I'm going to highlight 24 bases. So right here. Okay, right here. So I'm going to new feature it, and I'm going to call it just reverse primer. Okay. Double check that. Yep, 24 bases. And I'm going to, let's say, I'll just make it, I don't know, different color. How about green? Sure, why not? Okay, so here is the reverse primer. As you can see, it's at the end of the sequence, or I should say, it's downstream of the sequence that we want to amplify. So if you take this as a whole, we have there are 4 reverse primers located at the beginning and the end of our sequences. Now, what you want to do, of course, you want to place the order for the primer. So you want to, of course, copy the sequence. So instead of writing it down, what most people do is that just control and copy it. It's very simple and easier not to make a mistake that way. So what you want to do is just kind of highlight the beginning strand here, the top strand, and just Control C, copy, open a Word document. That's what I like to do, and boom, there we go. And this is the 5' to 3' forward primer. Right. What I like to do also is separate it in triplets. I don't know, just something I like to do. You don't have to do this, obviously, but to me this makes it much easier to read. And also, if I'm considering about frame shifts and such in cloning projects, this is a lot easier to deal with as well. Okay, so that's pretty simple. Okay, now for the reverse primer, remember, most websites, they want, and when you read it in literature, that when they show you 2 primers, the forward and reverse are in the same orientation. As in, both are from 5' to 3', written out that way. It's easy for the forward primer because it's just the top strand. But for the reverse primer, you need to write it in a way that's the reverse complement. Okay, I'll repeat that again. So you need to write it in the reverse complement direction. So if we're looking at the bottom strand here, right? So this is the 5' region right here where my cursor is. And here is the 3' region. So we want to write it from 5' to 3', and it's the complement of the top strand. And it's reversed because we're writing it this way instead of this way, right? So going from right to left instead of left to right. So this is a neat trick that you can do. All you have to do is actually just highlight the bottom, and then if you go, if you go this way, then it should automatically put it to the right orientation. So let's see if this works. Control copy, and the first, it should be TGA GCA. Let's see if that's true. Yep, TGA GCA. So it worked. So I'm just gonna do my thing here and separate everything out in triplets. And again, this is now 3' to 5' to 3', and this is the reverse primer. And yeah, and basically at this point you can go back, make sure to check the sequence is correct. But that's pretty much it. That's how you design very simple PCR primers, not taking consideration whether this is going to be used for cloning or not, because we're not inserting any restriction enzyme sites, or we're not doing this for homologous recombination, for example, back system or anything. So this is just, for example, you want to create primers and you want to amplify a segment, maybe for a confirmation. If someone gives you a plasmid or something and you're not sure if that target sequence is in there, you can download the sequence, design primers to target the specific sequence, and then you can run your PCR and then see, well, okay, is it actually there or not? So again, PCR is a very powerful tool, and I will make a video kind of going behind the theory of PCR and what it actually is, and a whole review on PCR basically. There's actually a lot of information, and I'm not going to get into that right now in this video. So I hope you enjoy this video, and I hope that you subscribe, because there'll be more to come later on. Thank you for your time, and I hope, again, everyone's research is going well. This is, well, this is it, I guess. I'm signing off.